Hey folks, thanks for checking in to Mr. Ulrich's Land of Biology. I am Mr. Ulrich. So far, we've been spending a lot of time talking about Mendel and factors and doing Punnett squares. And what we really need to do is start to put some modern molecular understanding of how DNA works into genetics. So this is going to be the first part of how we get from gene to protein, and we're going to be focusing on the step of transcription. Most of my high school biology students come to me knowing that DNA is the code for making organisms who they are, like my cute little son there. What we need to get to the bottom of is how those cells are made from the instructions that are in the DNA itself. And the first step, of course, is how do we get from DNA to proteins? And then how do we control those proteins, whether they're made or not, and where they're made, and how much they're made, and at what time they're made, and all that good stuff, to make the right cells, which, of course, can then organize to make the bodies that we know and love. The informational relationship between DNA and proteins is described, or was described, as the central dogma by James Watson of Watson, Crick, and Franklin fame. The three of them together kind of figured out the basic structure of DNA. And as they did this, they realized right away that with the base pairs lined up the way they did, that it naturally gave a mechanism by which a copy of that sequence, a perfect copy of that sequence, could be made through replication. What we're going to be talking about through the rest of this video is how that base pair sequence is then transcribed onto an RNA molecule, or into an RNA molecule, I should say, of the same basic sequence of bases. In this process of transcription. In the next video, we're going to talk about how that base sequence of RNA is converted into an amino acid sequence. We've talked already about how the amino acid sequence of the protein causes that protein shape through all the different interactions between the side chains of the amino acids. And it's from that shape of that protein that we're going to get the trait. So the shape of the protein is going to determine the protein's effect, whether it's there or not, and how much, and all that good stuff. And that's going to make the trait that is expressed. A number of years before Watson and Crick and Franklin were doing their work, Archibald Garrod, who was a doctor in England, was one of the first to make a connection between genes and disease. He was following the inheritance of metabolic disorder, and came up with this idea that because it's inherited, and we know that genes are passed on from generation to generation, that perhaps the metabolic disorder is a problem in the gene, and that each disease is going to have a particular gene product that is non-functional, that isn't working, or that comes from the lack of an enzyme. See, in a metabolic pathway, you're going to have several enzymes that catalyze the different reactions of that pathway, and each enzyme has a particular gene. Then what we'll see is if you knock out that enzyme, then you're going to be missing that metabolic pathway, and you'll have a particular expression of that disease. And if we knock out a different enzyme, then we might have a different expression of that disease with each different gene that is affected and knocked out. He was looking into a non-fatal inherited metabolic disease called alkaptonuria, which, interestingly enough, makes your urine turn black. Most of us can break down the material that's in the urine, and you don't see it. But these folks have a problem, have a mutation in a gene that codes for the enzyme that breaks it down. And there are several other inherited metabolic diseases that are associated with genes that aren't work. Albinism, not necessarily a disease, although it does cause some problems without being able to protect yourself from ultraviolet radiation. In the mid-20th century, about 100 years after Gregor Mendel was doing his work, Beadle and Tatum published a paper where they described a one gene, one enzyme hypothesis and proved it experimentally, that a pathway that would explain it, that states that each enzyme has a corresponding gene that codes for it. The experiment that Beadle and Tatum performed is kind of intricate and kind of elegant and kind of neat. It works. What they did is they took a bread mold, Neurospora, One of the cool things about Neurospora is it reproduces asexually, so we can get some asexual spores that have the same exact genes as their parents. Um, and the other thing that Neurospora can do is it can grow in what's called minimal medium. So minimal medium is a growth medium that has like sugar and salt and a vitamin called biotin. And from this vitamin, the Neurospora can synthesize, they have enzymes and pathways to synthesize all of the other nutrients that it needs. And so they took some of the spores from this Neurospora that could grow on minimal media, and they blasted the heck out of it with X-rays and ultraviolet lights, and they were trying to create mutations. And they took each one of these spores and they grew it up in complete medium to make sure that it could grow with all of the nutrients, that it was viable. And then they also put that and tried to grow it on the minimum medium that only had the biotin. And if it couldn't grow on the minimum medium, but could grow in the complete medium, that showed that it was what they called a nutritional mutant, that they had a mutation somewhere in one of the genes that coded for an enzyme in one of the metabolic pathways that converted the biotin to some nutrient that it wasn't getting now that it was on minimal medium. And now the trick is to find that mutant. And so what they had to do is they took spores from those nutritional mutants and they put it in all of these different minimum mediums that had one other added supplement to it. And if it grew on one of them that had that supplement, then they knew that what was missing in that was a gene that would convert the biotin into that missing nutrient. And indeed, it was arginine. That they had mutated just by random. Remember, they had no idea what they're doing. Nowadays, what we would do is we would just use CRISPR-Cas and snip out the actual gene that codes for the enzyme and cause what we call a knockout culture. We do this with mice and do it with all kinds of organisms. To see what the function of a gene is, we take it out and then see what happens to it. And that's kind of what Beetle and Tatum were doing. And they caused a mutation and showed that a mutation in that one gene caused a mutation in the enzyme that it coded for. One of the main players in this transcription game is RNA. RNA is very similar to DNA. It has a ribose sugar instead of deoxyribose sugar. It has nitrogenous bases in it, so it's a ribose sugar and a phosphate group and a nitrogenous base. And indeed, it has all of the same bases, except instead of thymine, it has uracil. So you're going to find uracil and adenine, or UA, are the complements, and C and G, just like in DNA. RNA exists very nicely single-stranded. And there's lots of different types of RNA: messenger RNA, transfer RNA, ribosomal RNA, silencing RNA, microRNA. There's all kinds of different RNAs now. It's a hotbed of research these days. Here's the problem. Eukaryotes keep our DNA safe and protected inside of the nucleus, instead of letting it get out into the chaotic and scary cytoplasm where there are endonucleases that will chew it up. The problem is that the code of the DNA, the base pair sequence, needs to be used out in the cytoplasm at the ribosome. And so what the cells do is they make a temporary, we transcribe, that means to literally copy over, we transcribe a temporary molecule that has the same sequence of bases that's disposable so that it can go out into the cytoplasm and hopefully get with a ribosome before it gets chewed up. And if it doesn't matter. I have the permanent code in the DNA. And this transcription makes what we call messenger RNA. Now, DNA is double-stranded, and only one strand is actually turned into messenger RNA, and that's called the template strand. The other strand is called the coding strand. It's the complement of the template strand, just like the RNA is the complement of the template strand. So it's going to have the same sequence, except for the A's and the U's. Where it comes unzipped on the DNA molecule, it's called the transcription bubble. And of course, all of this is catalyzed by enzymes. One of the enzymes is called RNA polymerase. And that's the one that puts each individual RNA nucleotide onto the growing RNA molecule. And it can only add those new nucleotides onto the 3' end of the molecule, just like in replication, the same problem that we ran into there. There are 3 different RNA polymerase enzymes. We don't have to memorize each one or even what they do. I'm just putting it out here for thoroughness sake. They're numbered, I believe, in the order in which they were discovered. So RNA polymerase 1 is the one that transcribes our RNA genes into our RNA to make ribosomes. R is for ribosomal RNA. Ribosomes are made out of RNA and a few proteins. RNA polymerase 2 is the one we were just talking about with making the messenger RNA. RNA polymerase 3 transcribes the transfer RNAs. Now, what allows these different RNAs to be made from different genes is that each RNA polymerase has a different promoter sequence. And so there's a sequence of bases that is encoded into the RNA polymerase that allows it to only attach to the DNA at a particular promoter sequence, so a particular sequence of bases. Now, these promoter sequences that allow the different RNA polymerases to know whether it's an rRNA gene that needs to be transcribed or a tRNA gene or an mRNA gene. These are called promoters, and it's the first hint that we have here that genes are a lot more than just the sequence of bases that gets converted into a particular polypeptide. There are promoter regions which come before the gene. The promoter region will also have what's called a TATA box at the beginning of it. It's part of what tells the enzymes that it is a promoter site. This is where RNA polymerases can bind and start to do their job, and there are other proteins that are involved that are going to allow the RNA polymerase to do its job, called transcription factors. There are other regions even further up, which are called enhancer regions. They're, like I said, they're kind of way upstream, and what these are going to do is they're going to allow those transcription factors to really bind well and do the job that they're supposed to do. And so we call them enhancer regions. They upregulate the gene. These transcription factors are suites or groups of proteins. They get incredibly complex, and we learn more and more about them every day, practically. And they're what helps these enzymes bind to the DNA and bind to the right part of the DNA so they get read. Some of them may be hormones that are taken into the cell. They may be the results of single transduction pathways in the cell that will turn on or turn off transcription. Some of them will bind to the RNA polymerase. Some of them will help the RNA polymerase bind to the DNA itself. Just like our language is directional, you have to start at one side of the sentence and move on the way through. DNA is also directional, so it can be read in the right order. RNA polymerase only works in one direction. It adds bases, RNA bases, onto the 3' end of the molecule, and so it's the 3' end that continues to grow. Our one gene for our one enzyme, one polypeptide, continues to grow as we are adding on promoter regions with their TATA boxes and enhancer regions that are even further up. Turns out that the actual expressed part of the gene isn't as continuous as we used to think it was. There are sections of it that are actually expressed. We call those exons. And then there are the parts that are in between the exons called introns. And this is essentially junk. It's never actually translated into a protein. It is transcribed. It's going to get snipped out, as you'll see here in a minute. And it's not always junk. Both the exons and the introns are first transcribed with the RNA polymerase into messenger RNA. And then it has to be, after it's transcribed, it has to be processed. Those parts need to be worked on before they're ready to be sent out into the chaotic cytoplasm. The primary transcript is before the introns get snipped out. We call that the pre-mRNA. And then we have some mRNA splicing where we take out those introns and chuck them. And then we put the exons back together again and make what's called the mature messenger RNA transcript. So we start out with this great big long strand of DNA, and from that, just the coding part, we make this primary messenger RNA transcript, which we call pre-mRNA. And then we splice out all of the introns and put it all back together again, and we get our mature mRNA transcript. Well, let's stop here for now with transcription. The next video will be about translation, and we'll get the rest of the story. In this video, we got the first part, remember, where we start with the DNA strand sequence of bases and how that gets converted into an RNA sequence, what RNA is, be it messenger RNA or transfer RNA or rRNA, depending on which RNA polymerase has the right promoter region. We talked about the expansion now of our concept of the gene to include those promoter regions and enhancer regions and exons and introns, and some of the post-transcriptional processing that needs to take place so that we can get that mature messenger RNA. If you have any questions, please feel free. Send me an email, drop them in the comments section, send me a note on Edpuzzle. Other than that, we'll see you in class.